waiting and trying to sort of figure out how we can best proceed with that. With the pandemic, unfortunately, that didn't help in getting together. But at least now we are in a position to do this virtually, and I think it was a good idea to do this anyway, even though the physical presence and interaction is still complicated and difficult. Hopefully, that will not be the case next year. I think this is an important time for genomics. I think genomics is becoming more and more relevant to all our activities, research and clinical. And we also see that by looking at also how the pandemic has evolved and how important even genomics is for the analysis of the viral genomes and their mutations, and how this has an impact on our ability to manage and to manage with the vaccines and beyond. But also, the variability, the human variability that leads to differential outcome, even in young people, due to specific variations that are predisposing individuals in ways that are different from the standard underlying conditions that have been identified as high risk. So we see in real time how genomics can actually transform our life. And I think there's many other ways that we all understand that could be transformative, but maybe not as acute as this pandemic. This happens, of course, the Swiss Genomic Forum happens normally in the environment of the Health 2030 Genome Center. It's organized by the Health 2030 Genome Center. And this is also an important time for the Genome Center itself, after almost 5 years of existence right now, that the Genome Center is basically moving to a new phase. It has actually been quite centrally involved in many of the efforts in Switzerland with respect to the pandemic, sequencing viral genomes, and participating in the Office of Public Health, big national project for the monitoring of these genomes and the mutations, in order to understand how this is going to impact Switzerland, but also participating in the international effort and the international monitoring. But at the same time, it's a big transition, because the Genome Center recently received an accreditation by the Swiss authorities to perform sequencing for clinical purposes. And in particular, it's a unique type of accreditation. It's not linked to any specific clinical protocol, but it's not a vertical accreditation that involves whole genome, whole exome sequencing, and RNA sequencing for all clinical purposes, as long as the protocol of collection of the sample and the protocol of interpretation of data is well defined in the clinical setting. So this basically puts the Genome Center in a new space that will allow for a stronger interaction and links with the clinical community in Switzerland, hopefully beyond that. And so the idea now is to move to the next phase, where we basically realize that potential. Of course, the clinical side is important, but also the ability to link with large international and national projects, and to facilitate through the genomics community of Switzerland and the international community the move to a sort of genomic medicine and more genomics informed medicine. So what we're going to do today, we're going to hear from exceptional speakers. It's great that we have this roster of speakers, both from sort of regional and local and European level, as well as from the sort of the other side of the Atlantic, that will give us their individual perspective, a quite diverse perspective on the different directions, topics, and dimensions that they're approaching the problem of genomics, information, ethics, how this is linked to drug development, but also some research projects that are sort of the cutting edge of the knowledge generation in this area. So I think it will be a very exciting meeting. Unfortunately, we cannot do it physically. It will be a lot more exciting. But I think the roster of speakers and the interactions and the questions, and at the end, the panel that we're going to have will probably be very rewarding, even in these difficult times. So I'd like to thank you again for your attendance, and thank you for being with us. As I said, hopefully next year with physical presence. And I'm going to turn over to our Chief Operations Officer of the Genome Center, Keith Harshman, who's going to be chairing the rest of this morning session and will introduce the speakers and manage and moderate questions and discussion. So thank you very much, and welcome, and have a good time for the rest of this day.
So hello, everyone. As Manolis said, my name is Keith Harshman. I'm the Chief Operations Officer here at the Health 2030 Genome Center. Welcome, everyone. Thank you for coming, and thank you for giving me the opportunity to be the moderator for the morning session. So we have 3 different presentations, 4 presenters, but 3 different presentations this morning, each lasting, including questions, roughly 35 minutes. So there'll be a period for questions at the end of each presentation. If you have a question, you can submit it through the chat that I think you'll be able to find along with the Zoom window. So you can submit those, and those will be read to the particular presenter at the end of their presentations. So I don't think there's anything else I need, no more administrative information that I need to provide. So I think we'll just begin right away with our first presentation, which is going to be given by Emma Hodcroft, who's coming to us from the Institute of Social and Preventive Medicine at the University of Bern. And she's going to speak to us about the application of practical phylogenetics for public health, realizing research in real time. So thank you very much for accepting our invitation to present.
So thank you very much, Keith, for that lovely introduction. And thank you, of course, for the invitation to come and speak. It's a real honor and a pleasure. And I actually wanted to just change my talk a little bit and talk more specifically about variants, because I think this is something that's really on everyone's mind right now. And it's something that obviously ties in particularly with how we can use genetics and how we can realize what we've accomplished this year in public health. So of course, what I mostly work on is using sequencing and phylogenetics. And just to kind of make sure that we're all on the same page, to go over really briefly how this works, when a pathogen moves through a population, it acquires mutations that are shown here as these colored diamonds. And then of course, some people, we will sample them and we'll sequence their viral genome. And when we do that, that allows us to actually see these mutations. And we can already see just from this really simple toy example that some viruses have more mutations in common, some have fewer in common. And this essentially lets us see how those viruses are related and lets us determine which ones are more closely and distantly related. And we can create a diagram that shows this relationships, which is what we call a phylogeny. It's essentially a virus family tree, and it uses those mutations to help put together sequences that are more closely related and put further away those that are more distantly related, which approximates kind of how a virus has moved and changed while it's been transmitting. But of course, as we've all heard in this pandemic, many mutations occur, and a lot of these don't necessarily impact how the virus works or how it transmits or the clinical outcome. So how do we identify when we have a new variant that we want to start paying attention to? Well, of course, one of the ways we look for new variants is we can look for epidemiological changes. So we might say, for example, okay, the cases have gone up in this particular area. But of course, cases can go up for many reasons, maybe super spreader events, changes in restrictions, seasonality, or behavior. And so we often also kind of link variants with mutations, and in particular, mutations in the spike protein, which of course is the one on the outside of the virus that's really important both for cell entry and for immune recognition. But of course, correlation does not necessarily equal causation. And so one of the biggest questions we have when we identify what might be a new variant of interest or variant of concern is trying to figure out, are the changes in the virus, are these mutations that we see actually the thing that's affecting things like transmission, immune evasion, or clinical outcome? And this can be really difficult to tell, particularly when a variant is first emerging. Our sampling is probably not complete, and we're trying to disentangle different types of effects that could be impacting what we're observing. But it's really important for us to try and do this. And critically, even if the virus changes themselves are not necessarily responsible for the epidemiological changes we're seeing, it can still be really important for us to look at this and understand how and why the virus is spreading in a way that's caught our attention. And I wanted to just highlight this a little bit by going back in time to last summer and looking at a variant that spread really well last summer. So this one was called, or it is still called, EU1, and it's here at the top of this phylogenetic tree in orange. It's defined by a mutation in the spike protein. And this animation shows from kind of the height of summer 2020 to the end of 2020, how this variant spread. You can see the orange growing, and it became the most dominant variant in Western Europe. And over all of Europe, it took up over 30% of the sequences in Europe. So it spread really well. And we can actually look at this in more detail by plotting the proportion of sequences from different European countries that were part of the EU1 cluster week on week. And when we do this, for example, Spain is here in red, and we can see straight away that we do think this variant started spreading in Spain. And from the beginning of June, it actually rose quite quickly to over 50% of the sequences coming in week on week in Spain. And it continued to rise through to the end of 2020. It was over 80% of the sequences. Now, of course, the other thing that was going on last summer is that after a pretty hard lockdown in the spring, borders started reopening in June. So Spain opened its borders in about the middle of June. And soon afterwards, unfortunately, we start to see EU1 samples popping up in different countries across Europe. And in general, the pattern we see is these rise, and then they start to plateau. Though there are, of course, differences in exactly how these patterns played out. And perhaps most importantly, we see differences in the frequency that EU1 ended up in different countries. In some countries, it's over 80% of the sequences in that country, like Spain and Ireland. In others, like Norway and in France, it really only hit about 20% of the sequences that were coming in were EU1. Now, of course, when I started looking at this last autumn in about September, we had this situation where cases were rising, the frequency of EU1 was rising, and we had a variant that was defined by a mutation in the spike protein, the one that we're all paying kind of the most attention to. And of course, first thoughts started to think about, is this a variant that's more transmissible? At this point, of course, the variants of concern had not yet been identified, but this is still what was on scientists' radar as something we were concerned about. But actually, the more we started to look at EU1, the more we became convinced that this actually wasn't a variant that was more transmissible, and that travel was actually what was really critical in allowing EU1 to become so prevalent. Now, I won't talk about it in this talk, but we have actually done some lab work as well. We've used pseudotyped lentiviruses. We've actually put that 222 mutation in there, and we haven't been able to find any significant differences that are caused by that mutation. So it doesn't seem to produce more viral particles, doesn't seem to change antibody binding. Certainly, it's nothing like the changes that we can see very clearly when we put, for example, 614 in there, or some of the mutations that are now associated with variants of concern. But how did we start looking into how travel might have affected EU1? Well, one thing is we can go back in time and look at what was going on last summer. So this is a plot showing the weekly incidence of SARS-CoV-2 across countries in Europe. So essentially kind of the case numbers. And you can see the lockdowns worked quite well, and case numbers were coming down really nicely through the end of spring, beginning of summer. And when borders reopened in June, they were really pretty much at a low. Unfortunately, though, soon after those borders opened, case numbers started to rise. And in particular, if we look at Spain, once again in red, you can see that Spain's cases started rising earlier and were soon higher than most other countries in Europe. There's a small bit of an exception here for Belgium in light orange, but overall, if you compare the red line to most of the other lines, cases were higher and kind of rose faster. Now, of course, at the same time, borders were reopening. So we can look at the departures from Spain, normalized by the size of the country. And it's very clear from here, you can see exactly when borders started opening and people started making their trips. And of course, these are departures, so returning back to their home countries. This peaked at the height of summer. It did start to come down a little bit. Some countries did start introducing some restrictions on travel to Spain, but in general, it really started to fall about the beginning of September, a more kind of traditional end of the holiday season, for example, when schools start restarting. But we were really lucky in that we were actually able to get even more detailed information than this. So we were actually able to get the average incidence not only across Spain, but in Spanish provinces. And on top of this, we were actually also able to get roaming data from people who traveled from different countries in Europe to Spain. So from these two things, we were able to know where people came from, exactly where they went within Spain, and how long they were there. and what the incidence was like in the place that they were while they were there. And then, of course, we can combine that with the local epidemic growth that was going on in their home country. So what was the epidemic doing when they got back from their holiday? And combining these three things, we can create a simple SIR model that allowed us to explore how EU1 frequencies may have changed over time. And this captures that dynamic of rising and then plateauing really well. What it doesn't capture as well is it doesn't actually estimate the number of EU1 cases so well. I won't go into the full details of how we looked into this and why we think this is. I gave another talk you can access at this tiny URL starting about 20 minutes in if you'd like to see me walk through all of the different aspects here. But essentially what this told us is that this suggests that travel-related activities were associated with more cases than just looking at the average incidence in provinces and travel data. So we're under predicting how many EU1 cases holidaymakers brought back from their holidays. And this really suggests that things like demographics, behavior, and variance in incidence really play an important role. So just as one brief example, for example, we know that even though we only had data to the province level, a lot of people go to very specific resorts. And the incidence at a resort is probably not the same, or it could not be the same as the province as a whole. So you might be in a much higher risk place being at that particular resort with, visitors from all over Europe. And of course, the activities you undertake also vary a lot by exactly where you go on holiday. And we know different countries actually go to different areas of Spain and seem to undertake different holiday activities. And those are going to vary a lot, going to vary your risk of getting SARS-CoV-2 and taking that home quite a lot. But all of this happened last summer, 2020. It's kind of old news. Why should we still be talking about EU1 now? Why is it still important? Well, though it might seem really obvious to us now with the variants of concern, this research really started before the variants of concern, but it highlighted, I think, really initially why tracking variants is so important. And in particular, it highlights the impact and the importance of travel. And this, I think, is definitely something that's relevant right now as we try and once again balance reopening with managing case numbers and taking countries on and putting them off different travel restriction lists with different travel requirements. And we can also look at what went wrong last summer and how we can do that better. So in particular, last summer, we allowed people to travel, many countries in Europe allowed people to travel to Spain, even though case numbers were rising quite quickly. We also didn't generally across Europe have testing available for travelers who returned back. It was really almost entirely honor-based quarantine, which may not have worked as well as we'd hoped. And then finally, what we would hope is if you did come back from holiday with SARS-CoV-2, a country's test and trace system would pick that up and stop that transmission chain from growing. But what we can see, of course, is that unfortunately, EU1 had plenty of opportunity to get a strong foothold in many countries when it came back with travelers and spread it extremely well. It also shows us the importance of taking into consideration things like travel volume. And this is particularly relevant with the variant, for example, at the moment, the variant that was found recently in India. We see many more introductions to the UK with its close ties to India than we see in many other countries in Europe. And this might explain, for example, at least some of the reason why case numbers have risen much faster there than in other places, at least initially. But finally, I think the really important message here is to keep in mind that not all rising frequencies are necessarily due to viral change. So we shouldn't underestimate the impact of human behavior. We definitely do have variants of concern right now that do have increased transmissibility, but we always want to be keeping in mind that it could be a combination, and often is probably a combination of human behavior and viral changes. And we have to start pulling that apart because there may still be things that we can do even with more transmissible variants, or ways that we can learn about how transmission is happening and then control those, whereas we can't necessarily control, how the virus is changing. But this, of course, is just one variant. And now we have multiple variants of concern spreading. This is a bit of an old chart, but it's showing the rise of V1 or B117 or alpha that was first identified in the UK. And you can see from the end of 2020 through March 2021, how this has come to absolutely dominate across Europe. And so I wanted to talk a little bit now about how we actually detect variants and how we learn about them and how we can do this better, because this certainly doesn't seem to be something that's going to come to an end very immediately. So first, let's talk about how we actually identify variants. There is definitely some algorithms and code that do this, but for a large part, this is relying on essentially people who stare at trees. So that would be people like me around the world. and local reports. So for example, doctors noticing interesting rises in cases, or scientists noticing that the frequency of a particular mutation has gone up recently in a particular area. Now, there's a lot more eyes on this now, thanks to the variants of concern. A lot of countries have invested and scientists are spending more time looking at this, but there's still little coordination. So for example, if you are to look at a particular tree, this is one I was looking at a few months ago. I was looking particularly at mutations at the 477 site, which is not one that we think is highly concerning, but it does show up in a few variants of interest and concern. And you can identify some clusters here that don't have names and aren't really recognized on any website. They might have a pango lineage, but that's about it. It can be really hard to know, is anyone actually looking at this? Has anyone noticed this? And you have to be careful how you do this. If I went on Twitter and said, hey, I've noticed a new cluster in Switzerland, has anyone heard about it? It will very quickly start spreading that there's a new cluster in Switzerland, which is not the question I asked. So we have to be able to do this in sensitive ways while still trying to connect with other scientists and find out if anyone else can help us learn about something that we've spotted, or maybe alert another country's system to saying, hey, we seem to see a thing, maybe you guys should look at it. But of course, to even look at these kinds of trees and to ask these kinds of questions requires that we have sequences. And if we look at the proportion of cases that have been sequenced in different countries, just in Europe, you can see there's a really big disparity here. We've got a few countries that are doing extremely well. Please note the y-axis changed just so that the graph can remain visible. But we've also got a lot of countries where we have a much less good idea of what's going on genetically in what's circulating. And this is just Europe, of course, but this represents areas where we don't necessarily have a good idea of what variants might be emerging or which variants are spreading, or that we have a really balanced picture of what an epidemic is like in a country. For example, maybe all of the samples are coming from one university or one town that may not represent that country as a whole. And of course, globally, the picture is even worse. This data is only from February 2021, but the message still holds true, that we have huge disparities in the number of sequences that we're getting from some countries, and particularly in countries where they can be having big outbreaks. And of course, this is leaving out a whole bunch of countries. These are at least countries that are sequencing, but there's a lot of countries where we really don't get regular sequencing updates from that country. And if a new variant appeared there, we would essentially just have to wait until it spread to a country where they are sequencing before we even knew it was something to be looking at. So we have real blind spots in our ability to monitor for variants, restricted by the types of sequencing that countries are able to do. But of course, overall, one more problem is that this is really a tidal wave of data. This is a flu tree, so influenza. And this was essentially the biggest pathogen sequence kind of group that we had prior to the pandemic, about 300,000 sequences gathered over decades. And of course, this is also something we know a lot about. We understand flu, we've studied it for years. We kind of know the parts of the tree and how to interpret them and which bits are giving us the information that we need. On the other hand, SARS-CoV-2, we now have almost 2 million sequences in just over a year and a half. That's absolutely unprecedented. And of course, we understand much less about it. There are many points on this tree that we're interested in knowing more about. We don't necessarily understand exactly how it's evolving or which exact mutations are the ones we want to look at. And we're getting this information in at unprecedented rates. And next strain, it's not uncommon for us to get over 10,000 new sequences a day. That would be a lifetime of work for most pathogens before the pandemic. And so another thing we have to consider is we need tools to handle this. No one expected to have 10,000 sequences coming in every day before the pandemic. If you'd written that into a funding proposal, you would have gotten laughed out of the room. But suddenly we do have challenges. We need to be able to sample and create trees that are useful for people, that focus on, their country, their town, or a variant that they're concerned about. And unfortunately, even though it's not very sexy, it's a bit mundane, data cleaning is a huge issue. We have to sort through errors in sequencing. At NextStrain, we do this every day with 2 students. They look through and look for date errors, look for sequencing errors. And of course, we have to standardize locations. There's a very Swiss example here, but this is a problem worldwide. There's often multiple names for locations that are put in that just don't make any sense. And this all has to be cleaned up so that we have a good idea of where samples are actually coming from. But once we do have these sequences and we've managed to use some of these tools, how do we actually learn about variants? One thing I just wanted to highlight is these are some screenshots of different variant prevalences in different countries in Europe over time. And this is from my website, covariants.org, that I manage. And it's a little bit old, but what I really want to highlight here is that not all of these are variants of concern. In fact, variants of concern are just the ones in red at the end. I wanted to show older screenshots because now the red is kind of everywhere. But this is over the summer and autumn of 2020. And you can see there's a real tapestry here. There's a lot of different variants that were circulating in different countries at different times. And even though these aren't variants of concern, we really know very little about most of these. EU1 is here in orange, but it's about the only one that's been really, really closely looked at. Now, of course, a lot of this is just going to be stochastic. Variants will rise and fall that have nothing to do with the variant themselves. But I also don't think there's nothing to learn from non-variant of concern variants, just as is the case with EU1. But we're moving so fast, we have so much to do and so much data to parse that we've totally missed the opportunities that might have come, what we might have learned from some of these variants in the past. The other thing we have to handle is that we have an unprecedented amount of work coming in. We have information from modeling and experiments looking at spike structure, looking at expression, looking at ACE2 binding, looking at antibody binding. We have amazing epidemiological and clinical studies, and of course, phylogenetics, what I do. But how do we actually bring this together and make it actionable? If you find a new variant or new mutation, all of this information is out there on blogs, on Twitter, on preprints, on publications. But actually pulling that together so that you have a good idea of the implications of what you might have found is actually quite difficult. Coordinating that amazing amount of information is an ongoing challenge, and it gets harder every day with the more and more amazing work we keep doing. And of course, the big caveat on that is that we are also trying to do that while working as quickly as possible, already operating at max capacity. So how do we improve? We're in this amazing situation where we have more data than we ever imagined, and we're drowning in it. And that's a privilege. This is not necessarily a problem we've had in the past, but it would certainly be better if we were swimming in it rather than drowning. We have more sequences, more data, but also more urgency than ever before. And unfortunately, our systems and tools simply weren't made for this. No one saw this coming. We've done an amazing job repurposing things we have, improving things in tools and analysis, but we still want to make sure we're looking forward, because the next time this happens, or even if it's not a pandemic, but an epidemic, we want to make sure that we haven't underestimated what we need to use what we can generate most effectively, particularly because sequencing is only going to become more ubiquitous. So how can we go forward? First of all, for detecting things like variants, number one, we need more sequencing. We need more equitable sequencing access worldwide. We have too many blind spots, and these are increasingly going to happen in places where SARS-CoV-2 continues to circulate, where vaccines aren't as available. The more sequencing we get, this will not only pay off for SARS-CoV-2, but it will also mean that we have a global surveillance network that can be used next time we get an epidemic or a pandemic that's worrying, and can be harnessed by those countries to then dedicate to whatever local pathogens might make a difference to their own health and security as a country. We also need to make sure we're investing in tools so we can create the starting points that we need through data subsampling, making trees, and data cleaning, and we can effectively use the huge volumes that we're getting in. We also, I think, need to ask ourselves who should be sequencing. A lot of the sequences in this pandemic have been generated by academics, but that can also mean that there are restrictions and concerns around using them because academics need to be recognized through publications. In Switzerland, we've just introduced the Genome Surveillance project, which means the government will fund the sequences and they'll be available openly. If we want to think about sequencing in the future, it's no longer purely an academic exercise. Of course, it will still underpin tons of research and academic work, but it's going to be more of a public health tool. So we need to maybe rethink about how we're going to actually fund it and use it. When it comes to learning about variants, we need to invest in expertise so that we can start swimming in this huge amount of data that we have and make the best use of it and not overpass opportunities to learn, even if they aren't necessarily the things that are most concerning, to make sure we're grabbing every opportunity to learn what we can. And we need better ways to combine the data that we are generating so that scientists can access it more quickly and make it more actionable. And then finally, we want to think about communication. In particular, I think it would really aid a lot of scientists working on SARS-CoV-2 to be able to connect to other scientists in a way that's not necessarily on something very public like Twitter, but where they can discuss issues. In the US, they have a Slack group called Spheres that I'm in as part of NextStrain, but any scientist working on SARS-CoV-2 is part of this. And it's a great way that people can just ping other scientists and say, hey, our local health department wants this. Do you have a format for this file that I can use? Or, hey, we're having trouble with sequencing. Can anyone give us a hand? Something like this in Europe could be really powerful, particularly because a lot of the laws and concerns are really similar to researchers here. And we're probably missing an opportunity to better share our information about tracking and identifying variants in ways that aren't necessarily, putting something up on Twitter where it can be misinterpreted, particularly because sometimes when we find a variant, I'd like to say they're variants of should we worry about this? Talking to other scientists helps us to disentangle that. And it can also help us translate what we're doing to be more actionable, to make better connections with our governments and with our health departments, so that all the science we're generating can actually make a difference to the public and to our outcomes as a global community, which is obviously the end bottom line, is that we want to make differences in how this pandemic is happening globally. So with that, I'd just like to thank everyone who was involved in the EU1 study, which was an amazing collaborative project and should be out hopefully next week in Nature. And thank you all for listening. I'm happy to take questions and remind you that it will be much better in the future if we can move from drowning in our data to swimming in it. Thank you very much.
All right, thank you, Emma, very much for for your presentation. As I said earlier, people can submit questions to the speakers via the chat, the Zoom chat. My name is Keith Harshman, so if you send them to me, I'll read them out to the speakers.
It might still be too early for people to have thought up questions so quickly, but I guess while we're maybe waiting for a question or two, though of course it's never bad to be early in a seminar, I would say. Another thing that I would touch on is the connections that I think we should be building between academia and public health. A lot of the work in this pandemic has happened because of academia's being kind of seconded into public health. And that's fantastic in terms of it's allowed us to use groundbreaking research in public health. But a lot of these bridges are pretty tenuous. They come on top of professors who are teaching, who are still guiding students and doing their own research. And we want to make sure this is sustainable. Whereas at the moment, I think a lot of it is done out of goodwill, but there's a real possibility of burning academics out and leaving them kind of unrewarded in academia, where of course we tend to recognize talks and publications, but not necessarily public service in terms of helping out with public health. So I think exploring how to build better bridges is another thing that would be really great to do going forward.
Keith, since there's no question yet, I would like to ask a question, if that's okay.
Of course.
Yes. So Kelly, I think that it was great to have this presentation, especially because it's such an important and timely presentation for all of us. And the worries about the different strains and mutations and the dynamics. What I would like, I mean, you alluded to that a couple of times in your presentation, but I would like to get sort of a perspective on a clear and more sort of specific perspective on how easy it is actually to differentiate between sort of chance events and increases of a certain variant of SARS-CoV-2, for example. Now it's like the Indian one or whatever it's called, like the Delta, I think, right? That's the new nomenclature. And to differentiate between just kind of a dynamic that happens by chance, given the spread of the virus and the different behaviors and so many things that we simply don't know, versus a concrete conclusion that it's actually more transmissible and it's by that component. Like, for example, one of the things that worries me about that Delta variant is that we do see an increase in the UK, but is that among the same people that we don't see an increase for the Alpha, which is the UK strain? I'm just a little bit worried that there's like two different communities that are probably differentially vaccinated. And yes, we measure more cases in the UK, but is that really evidence that the differential, let's say the opposite trend where the Alpha goes down and the Delta goes up on its own without taking social aspects, distribution of vaccines, minorities, could we just say that it's more transmissible just because one goes up and the other one down in the same country? And so how do we solve that problem if there's all these discrepancies and differential kind of, sub-presence within the society?
No, so I think that's an absolutely on point question. And it's the hard one that we're trying to answer, every other week right now. And I think that you've raised some really important points. So we know, for example, that unfortunately, ethnic minority communities in the UK are less likely to be vaccinated. and that those are probably the communities where many arrivals from India, that might have been carrying this variant arrived back and lived with, have extended family with. And so there's certainly, questions there about, is this just spreading, really well through, unfortunately, unvaccinated pockets of the population that might not necessarily reflect how this would spread in a more general community. At the same time, we do see some worrying trends in the transmission rate, even when you try and control for these things. But it's really hard to pick these things apart completely. It's really, really difficult. And I think that that's one reason why being able to look at these kinds of trends in different countries is very important, because you might be able to start picking apart, maybe outside of the UK, other countries that maybe don't have such strong connections to India, you might be able to say a little bit more, okay, this might be in the more general population, or our vaccinations are, maybe just more kind of homogeneous across the population. But we also just have problems even in, for example, with this variant, when it first popped up in India, if you plotted all of the sequences coming from India, it looked like the Delta variant was taking over Alpha. So 617 was taking over B117. But if you then split it out by region of India, it turned out that there was actually really different sequencing patterns happening in different areas, and that it wasn't such a clear pattern when you started to control for that. Right. But of course, availability of data is a huge bottleneck in terms of what you can do and how well you can control for that, particularly in countries where the sequencing is being really diverted to a variant that might have just appeared. So I think this is a huge question, and it's ongoing, and we've gotten it a little bit wrong. We can all think of variants that made headlines, in the last 6 months that we haven't heard about since. So it's not always an easy question to answer at all. And I think we have to find that balance between knowing when we want to take action and not alerting the public so many times that they essentially stop listening.
Exactly. Yeah.
Okay, so we have a question from the audience. So first of all, a compliment you, a great talk, and then asks, what's the bottleneck in terms of data availability? I get the feeling that high resolution local case or death numbers are lacking too, maybe even more than good spatial coverage of available sequences.
Yeah, so this is a great question. And this is certainly something that is perhaps a challenge for Western countries that have managed to really up the number of sequences that they're generating, where number of sequences is maybe not a problem, but being able to attach meaningful data would allow us to make more from those sequences. And in particular, something that countries are getting really interested in right now is making sure they know the vaccination status of individuals, because of course, no vaccination is perfect. We're going to see breakthrough infections. That alone is not necessarily something we need to get worked up about, but we definitely want to be monitoring to see if we see, everyone that had a vaccine and got infected, they've all got the same mutation. That would be kind of a bad sign. We want to be able to pick that up early. If we don't know vaccination status, we wouldn't necessarily be able to find that pattern. And the same for many of the other demographics, socioeconomic status or age or gender. So it is a balance here. And I think different countries should be trying to, look at what they can do and how to set goals that kind of reflect that. In a lot of places in the world, just getting more sequences of any type would be helpful. In other places, it's trying to make sure that they aren't coming from, say, the capital city or one small region that has, the university with the sequencer, because we would like a better geographical spatial coverage. But there are now a lot of countries, particularly in the West, in the US and Europe, where our next challenge is how do we link this up with data more effectively? And I think that is something that we want to be thinking about going forward, particularly as as case numbers go down. We want to make sure that we're making the most use out of the kind of decreasing number of sequences that we as a country are generating.
Another question from Carolina Borges. So are there any initiatives to create a system of global governance to sequence new variants?
So that's a great question. I'm not sure if there's something in progress, but we have something like this for flu. So there's a WHO kind of helps to run alongside, of course, many global partners like the CDC and the ECDC, a network that tries to gather diverse samples of flu for each flu season, or in more subtropical in tropical regions, throughout the year. And this helps us to be confident that we're getting, a good spread of flu each and every year that we obviously use to try and help predict what will be the next flu and how to make the next vaccine. I think we could build on this for SARS-CoV-2, but we're still going to have to do some building there, because with flu, we still don't necessarily get samples from, for example, most of Africa. We would want to get these for SARS-CoV-2, but it's a starting point. So I think it's something that we could work with and make better.
Another question from Jerome Thomas. Do you think there should be an efficient way to coordinate and make bridges between academic labs, for example, or a national center for generating the sequencing data? And who should be lobbying for this?
So I'd love to see an easier way for academics to connect. I'm not necessarily even sure this needs to be so formalized, because I think academics are pretty good, or at least in this pandemic, have been pretty good at trying to connect. I think it's just making this easier, because if you have to go, if you see something interesting in a tree, but it doesn't look like it's kind of headline worthy, maybe this takes, Googling the sequence, finding out a lab, Googling an email address, and you're kind of like, well, I've got 6 million things to do today. This is not top of my priority list. If we have, for example, Slacks or forums or other groups where it's easy to get in touch with the people in that country that you should shoot this email to, then you lower that bar so that everyone can kind of communicate maybe something that they've seen that's interesting. What I would really like to see actually is to see more long-term bridges built in public health and academia. We've in this pandemic, we've really had to move over what was essentially used mostly for research. A lot of tools that were really research tools are now being used as public health tools, and that's fantastic. And we can never expect that day-to-day public health is not going to, be continually adopting the breakneck newest technology from academia. But we want to make sure that we've got something there that isn't trying to make new connections every time we might need something from academia and public health. And I'm very conscious of the fact that, in academia, we don't generally recognize formally this type of work, helping out the government or your local canton or something. You don't necessarily put that on your CV and get credit for that like it's a paper. And we don't want to disincentivize that, well, listen, I worked in the pandemic, last time, and I really feel like it didn't help my career at all. So this time I'm going to sit it out. We don't want people doing that. So we need to think of more sustainable ways to kind of reward and recognize service that is for the greater good, even if not necessarily for the CV good or the career good. We should make it for the CV good. Oh, you're muted, Keith.
Yeah, there we go. So a question about the spread of variants. So what about the spread of variants between different age groups? For example, children versus adults. Are there different variants spreading?
So that's a great question. I don't think that we've seen any evidence that this is a really strong thing, that like this is an intrinsic property of a variant. This variant really infects just one age group over another. It's mostly been probably because of more human behavior type things. So for example, in the UK, in one of the autumn lockdowns, the only thing that was left open was schools. And so then you perhaps not so unexpectedly see more transmission happening in schools. That doesn't necessarily mean that children are transmitting it more. It just means that that's the only kind of place where people are left to mix. Unfortunately, for somewhere like the UK, we have a lot of data where we can pick these things apart. But for a lot of countries, we don't necessarily have this data. So we have to be really careful when we see age type associations, that this isn't an artifact of sampling or of restrictions like, all the schools are open or all the schools are closed or schools are banned from contact tracing. So many different things that have to be taken into account. But in general, I would say we haven't seen any evidence that variants are only spreading in one age group or another.
Okay, so there's no more questions coming in. I think we're keeping which is not necessarily a bad thing. We're keeping on this. No, as a moderator, I'm responsible for keeping. So I'll thank you again for giving a really great talk, and then we can move on to our next speaker. Okay, who is Mike Inouye, who's at the Department of Public Health, coming to us from the Department of Public Health and Primary Care, which is part of the School of Clinical Medicine at Cambridge University. So the title of his talk is Polygenic Scores, Potential for Clinical Utility and Target Identification.
Great. Thanks, Keith, for that introduction, and thanks for allowing me to speak today at your lovely symposium. So it's really great to be following on from Emma. I mean, it's going to be a little bit of a shift here now between viral genomes and human genomes, but hopefully not too far for a lot of people. So so I'm going to be talking today about polygenic risk scores, probably something that many people here have heard about. They are rooted in the human genome sequencing project, which completed in roughly 2003. And from there, the evolution of polygenic scores, it really sort of came out of a lot of the international projects which followed on from that. And those include the international HapMap projects, 1, 2, and 3, the 1,000 Genomes project, and the kind of era of genome-wide association studies, or GWAS, which have mapped a lot of the phenotypic variation and sort of linked that to the genetic variation that we see across individuals and in the human genome. So really so that basically means that for common diseases, what we've really observed is that there's mostly kind of a relatively simple model where the genome here, where it's depicted on the left, so these are obviously all the chromosomes, contain a lot of risk variants, and those are denoted here by V's, which either increase or decrease risk. And obviously, as many people know, this is in contrast to rare monogenic variation, where a lot of times it's just one variant, which causes at a very high penetrance the disease. So common diseases, it's a lot of variants sort of sprinkled around. So using the sequencing of the human genome and the maps of the disease variants that genome-wide association studies gave us, we can combine all of these risk variants and protective variants into one genetic score, or one number per individual that quantifies their personal risk of someday having that disease. And we call these polygenic risk scores, or PRSs, which you'll hear me say throughout the talk. And they go by many names: genomic risk scores, polygenic scores, that sort of thing. But they all kind of mean the same thing. Now, it's important to stress here that DNA is not destiny, and we're talking about risk. So there's a wide distribution of risk in the population. Some people have low scores and low risk. Many have medium scores and roughly average risk, while others have high scores and high risk. And a small number have extremely high risk, say 3 or even 7-fold the average. And importantly, for nearly all common diseases, whether it's heart disease or strokes, diabetes, cancers, or depression, we do not currently use this genetic information to guide how we prevent or treat disease. It's currently hidden individuals it's currently hidden information from both individuals and the healthcare system. So in terms of risk prediction, polygenic scores have been called the first risk factor. A polygenic score itself is fixed after conception, and it is not really subject to the spatiotemporal fluctuations that we might see with other sorts of things that we study, like blood pressure or cholesterol levels, or that sort of thing, or body mass index. So together with sex, also genetically determined, a polygenic score can be viewed as the baseline hazard on which everything else to the individual is forward in time. And this is already a prism through which we view monogenic disease risk, such as that of mutations for familial hypercholesterolemia, which predispose to coronary artery disease. So as Ami Kira and colleagues have shown, individuals in the tail of a polygenic score distribution for coronary artery disease can have levels of risk equivalent to a monogenic mutation, and polygenic scores in the tail tend to be more common in a population than the monogenic mutation. So, as such, it seems like polygenic risk has a clinical role. And a key question that then arises is what action to take. So these include, for the individual, modifying their behavior. And you'll hear later on from Samali Rapati some excellent work that's been coming out of the Finnish Institute of Molecular Medicine, where they've developed a really exciting tool like CardioCompass, which helps facilitate behavior change. Other interventions can include changes to screening, such as screening initiation or frequency, independent of or in combination with therapeutic targeting. And that is, who you might actually treat with actually use pharmacology on. And for some diseases, we can use polygenic scores to enhance existing risk prediction models. And that's really exemplified by, at the moment, exemplified by breast cancer and the Boadicea/CanRisk polygenic score tool. So for the use of polygenic scores to enhance in existing risk models, my lab has done some work in the area of cardiovascular disease, much of which in collaboration with Emmanouil Ripati, who you'll hear from later. So using a particular approach called metascoring, we've published polygenic risk scores for coronary artery disease and ischemic stroke, where these PRSs outperform previous polygenic scores, as well as any single conventional risk factor, except for example, polygenic risk of stroke and versus blood pressure. So blood pressure is quite a strong risk factor for stroke. In each case, as expected, the best risk models integrated polygenic scores and conventional risk factors. So that's really something that you see all the time in common diseases. We're really not looking or trying to utilize polygenic scores on their own. We're always combining them with other risk factors to give us maximal predictive power. However, so at least for cardiovascular diseases, a major challenge is that we actually have to take these models and not just predict individual manifestations like coronary artery disease, which we have quite a bit of predictive power to do. We have to predict more heterogeneous phenotypes like cardiovascular disease overall. And that's because the therapy is really the same. So like cholesterol lowering medications or antihypertensives. So as such, we've really decided to try and assess the clinical utility of these PRSs to enhance conventional risk models and guide statin treatment. So cholesterol lowering medication for cardiovascular disease. So this is published in a recent paper in PLOS Medicine a few months ago. Overall, we found that even modest increases in C statistics, say 1 to 2 or 2.5%, can actually translate into meaningful clinical benefits. The polygenic scores correctly increased or decreased 10-year risk of cardiovascular disease for about 22% of participants. And notably, we found that an additional 7% of cardiovascular disease events may be prevented when including these polygenic scores in conventional risk factor models and following national guidelines. So in this case, we were looking at the NICE guidelines from the UK. So what does that really mean? So using UK Office of National Statistics data, ONS data, if we do a quick back of the envelope calculation to extrapolate to UK-wide this PRS guided statin usage, it may help prevent an additional 20,000 or so cardiovascular disease events per 10 years. That's a moderate improvement and a potentially a large number of events depending on how you look at it. But really it should be at this stage taken with a grain of salt, as there's wide confidence intervals on that in lieu of more rigorous modeling. And notably, overall, this is really just considering polygenic scores alongside conventional risk factors. We aren't yet modeling the PRS first approach, so taking advantage of the fact that this information is available very early in life. So PRSs may also have utility in supporting diagnosis. This is particularly promising for autoimmune diseases. In type 1 diabetes, people like Richard Oram and colleagues have designed and validated a polygenic score which has an AUC of about 0.92. And it shows promise in guided selection of newborns for autoantibody screening and the classification, as well as the classification of type 1 and type 2 diabetes in adulthood. So the latter is being important to avoid incorrect treatments, as well as reduce medical costs and morbidity. In celiac disease, multiple studies have shown that a polygenic score can improve upon traditional HLA typing. Back in 2013 or so, we showed that the machine learning approaches in particular can be pretty efficient at prediction for highly MHC dependent disease architectures. And this was certainly the case in celiac disease, where scores externally validated across 4 different data sets with AUCs of between 0.87 and 0.9, so pretty high. So similar to type 1 diabetes, polygenic scores for celiac disease have the potential to replace HLA typing and combine with or guide serology. In celiac, it's particularly important to minimize things like gluten challenges, which is always a costly and painful exercise, and the small bowel biopsies that end up following those gluten challenges. More recently, in juvenile idiopathic arthritis, a common cause of disability in children, we've trained polygenic scores, again, using machine learning and regularization to predict both JIA and its subtypes. And a polygenic score for JIA in particular may have substantial clinical utility because diagnosis of JIA is currently purely clinical. There are no accurate molecular tests to support it. So interestingly, the JIA subtypes where we had the most predictive power corresponded to the oligoarticular subtype, which is the most common, and the enthesitis-related JIA, which has the longest waiting time to secure a diagnosis, a median of roughly 11 months before a pediatric rheumatologist can actually diagnose. So, I've given you a few examples here in cardiovascular disease and autoimmune disease of areas where polygenic scores have potential clinical utility. And really, while they show a lot of promise for improving healthcare, there's still many challenges for polygenic scores. I'll go over a few here, but there's a whole laundry list of things that still need to be sorted out before they're utilized in clinical practice. So one of the major challenges has historically been the poor and variable reporting of polygenic risk score research studies. And you can see here on the slide really how that varies from paper to paper. You're looking at discrimination statistics like AUCs, AUPRCs, then onto like more modeling based metrics like hazard ratios, R-squareds, odds ratios, et cetera. And it's very hard to compare different studies, and even to compare two different studies that are done on the same phenotype. So, and one of the real challenges is actually that creates not just an ability to interpret, but because people didn't share their polygenic scores, it also inhibited their ability to, other people's ability to reproduce that research. So what we did was we set out to create an open repository called the Polygenic Score Catalog, so pgscatalog.org. And so you can go there now and sort of browse around. And what it contains is the scoring information and relevant metadata in order to apply and evaluate polygenic scores correctly, or at least more rigorously than it was. So the kind of aim here is that we catalog and provide all published polygenic scores, as well as provide systematic benchmarking. Now we're probably not going to get to that, right? Because polygenic scores are not. It's very hard to chase down all the polygenic scores that have been published and to provide them. So, but we're not doing half bad. So we've got probably almost 800 or so polygenic scores at this stage. The slide's just a little bit old. Over 200 traits and approaching 200 publications now. So you can read a lot more about the Polygenic Score Catalog and its development in Nature Genetics from earlier this year, or you can give us a follow on Twitter and pretty active Twitter account. I should also note that this is the work of a very talented postdoc, Sam Lambert, in close collaboration with the European Bioinformatics Institute in Hingston, HGR UK, and the Baker Heart and Diabetes Institute in Melbourne, Australia, as well as Jackie MacArthur, who's the lead on the EBI side, who's provided a lot of the infrastructure and know-how to get the PGS catalog started. So the PGS catalog contains a lot of critical information about each polygenic score. You can browse polygenic scores. You can look at browse traits as well. So when you click on the polygenic score, it comes up with a page that provides you with a lot of information. So not just about the phenotype that it's trained on, but stuff like the genome build, stuff about the modeling, what the parameterization of the model was, the ancestry distribution of the training, testing, and evaluation data, as well as the usual stuff like where it was published, et cetera, and also a link to actually just download the score as well, which we find is really a lot of times some of the most important stuff that people come to the catalog for. We also include a section on performance metrics, and this is so this we provide with quite a strong disclaimer there at the top, which is really saying what I said. You have to be very careful when you compare performance of polygenic scores across studies, because you're looking at different individuals, a lot of times different traits, different modeling approaches, different metrics, all that sort of thing. But we provide that here. So one of the things that we're going to be releasing here pretty soon, and we're just in the middle of assembling all the analyses for, is a polygenic score benchmarking. And this is really trying to address the question of, for a particular trait, what's the polygenic score that performs best in a given ancestry? This is something that we get asked, at least once weekly. And we're really trying to put together a functionality here as part of the PGS catalog that gives that information to the user. And this is a little sneak peek of what it's going to look like. So this is kind of one of the main interface, or this will be one of the main plots basically. So along here on the x-axis, you've got all the published or a lot of the published polygenic scores for coronary artery disease. Along the y-axis, you've got the odds ratio per standard deviation of that polygenic score in predicting coronary artery disease. And then in different colors, you've got different ancestries, whether it's European, South Asian, African ancestry. And you can see the per SD odds ratio of each polygenic score, kind of how it varies across ancestry. So a lot of times you'll see, right, the attenuation of a polygenic score effect as you move further and further away from European ancestries. You can visualize that here for the same samples, and for different scores for the same samples. And you can also see the kind of the progression, I guess, of performance across different polygenic scores, and which ones you might want to utilize yourself, right? So there on the far left, those on the far left will perform better than those on the far right, right? And that information is not necessarily apparent when you start looking at all these different published studies, right? You have to have one data set and compare directly and benchmark it. So, so hopefully that gives you a good idea of a useful tool that you yourselves can immediately start using, as well as some of the stuff that's on its way. So for polygenic scores to make their way into the clinic, there's another key challenge, and that's standards. So while best practice is evolving, it's really difficult to pin down the best approach for a particular task. But really one thing we can do right now, apart from best practices, is to improve our reporting. And this is something that we linked up with the NIH ClinGen Complex Disease Working Group, Jen Wojcik, Hannah Wand, and various other PRS experts. And we recently performed a review of 30 representative PRS publications and found that there was a really glaring lack of reporting for basic elements of the study. And these included stuff like statistical model or statistical validation, or information on the statistical validation, ancestry of the data, data transparency and availability. Roughly a third of polygenic score papers did not provide the variance and weights for the published polygenic score. And this collaboration really came up with the polygenic risk score reporting standard, or PRS-RS. That's a set of about 20 criteria that are needed to establish a polygenic score's analytic validity, transparency, and reproducibility. So hopefully thus facilitating its clinical translation. In parallel to the PRS-RS, there is an international peak body. There are international peak bodies, such as the International Common Disease Alliance, ICDA, which are working on various aspects that are integral to making polygenic scores available, equitable, ethical, and translatable for public and patient care. And I'm just flagging this because the ICDA PRS task force is really just getting off the ground, but we hope is going to be leading the way in a lot of these different areas. Okay, so I've talked about a lot about polygenic scores and potential use, near-term use in the clinic in order to, aid screening, risk prediction, that sort of thing. But polygenic scores also potentially have a model and a role in understanding the underlying biology, and that's something that we've been focusing on recently. So you can imagine here what I call kind of a systems genomics model for disease, where you've got, a person who has polygenic scores calculated for all sorts of different diseases, and we can select one here, say like heart attack, and really what happens is as they go through life is that they'll and this sort of network that I give here is like a protein-protein interaction network, is that as they progress through their life course, they'll start being sort of aberrant levels of a particular protein or particular proteins in that network. And those sort of aberrant levels will start accumulating and accumulating, where, say later on in life, age 70 or potentially even earlier, if they're at high polygenic risk, age 50, let's say, they have a heart attack. And we want to get an idea of, what role does polygenic risk play in this process? Does it speed it up? Does it slow it down? Does it change the molecules that are involved? Different PPI networks. That's something that we want to study. So what we've done, there's a little bit of a busy slide, apologies for that, is we've started looking at multi-omic cohorts. That is cohorts which have a fair sample size and which have the genotyping available to calculate polygenic scores, and which have the molecular data available to start looking at the downstream molecular consequences of high versus low polygenic risk. And here's an exemplar in the interval cohort, which crucially is an asymptomatic cohort. It's relatively healthy. These are blood donors. And so you're not really, you don't have a lot of reverse, confounding by reverse causality. So we decided to focus on 5 different cardiometabolic diseases: coronary artery disease, stroke, type 2 diabetes, chronic kidney disease, and so on, and look at the relationship between the polygenic score and individual proteins in this cohort, and then to compare, what happened, or to look at what happened to these individuals further downstream by looking at linked electronic health records. And we had about 7.5 years or so on average of follow-up health records. So, as you can sort of see there on the right, we're basically correlating protein levels with polygenic scores, and then protein levels with incident disease, and then doing a mediation analysis where we're trying to estimate the amount of variation in disease risk that seems to flow, that is attributable to the polygenic risk, but which also flows through individual proteins. And we can do this because we've got one cohort with all of this available data. We can look at sort of downstream things like for proteins that we think are mediators, are they targeted by therapeutics? Is there corroborating evidence for these proteins being involved in disease, et cetera, et cetera. So when we do that, when we correlate the polygenic scores with the protein levels, we find a fair number of correlations, roughly 49, and we can look at the distribution of those correlations across diseases. They tended to be with type 2 diabetes and coronary artery disease and CKD, maybe one with ischemic stroke. In particular, we were able to look at individual proteins, which seems to correlate with polygenic risk of multiple different diseases. So, for example, sex hormone binding globulin (SHBG) was associated with type 2 diabetes, coronary artery disease, ischemic stroke, but also so we could look at those which are associated with multiple diseases, but also ones which were specific to disease. So we looked a lot at the robustness of these associations. So there's various confounders that could be, factoring in here, whether it's, technical factors, seasonality, sex, age, et cetera, et cetera. They seem to be very robust to all the various factors that we had measured. And we also looked at, cis QTLs as well to these proteins to make sure that we're actually looking at a polygenic association with that protein level. So that is sort of here in this sort of purple panel here. And there were a few proteins which were their polygenic risk associations were attenuated by known cis pQTLs. But by and large, these associations were truly polygenic. roughly taking about 10 to 20% of the genome needs to be removed to actually ablate the association. And very reassuringly, we recovered known biology. So for example, C-statin C and beta-2 microglobulin, they're known chronic kidney disease biomarkers. And of course, ApoE and coronary artery disease, and FBP1, the target of metformin, and type 2 diabetes. So we found things were known. We found things that that are currently sort of not a lot of literature support, and perhaps aren't so studied as these are. So that was really heartening that we found a lot of robust associations. We can take all of these associations forward to actually look at the mediation. So, as I said, are these proteins actually showing, is there evidence for them as being mediators between the polygenic risk of coronary artery disease, for example, and incident coronary artery disease in the same cohort? And when we did that, we found, we thankfully found, I wouldn't say like, smoking gun evidence, but I think, and that's partially due to sample size. But we found broad consistency between the estimated effects of the protein levels, so the PGS on the protein levels, and the protein levels on incident disease, and that's being shown here on the bottom left. So really kind of, I would say, kind of surprising consistency between those effects. And when we actually test for the mediation, we find a lot of p-values which are statistically significant across these 49 associations, none of which are passing like a Bonferroni correction, but there's clearly an enrichment of low p-values. And in particular, we find things like ApoE, obviously, for myocardial infarction, which we know, and a lot of potential mediators for type 2 diabetes. Which explain and this is still something, an area that we're trying to look for. The proteins themselves explain roughly 7% of the polygenic risk of coronary artery disease on the incident myocardial infarction, and similarly, the 16 or so proteins explain roughly 27% of the polygenic risk for type 2 diabetes on incident type 2 diabetes in this cohort. So we also found consistency of the putative polygenic risk mediators with pharmacological interventions. So for example, metformin again. And also because we can look at the approved or the drugs that have made their way through trials, and some of which are actually on the market, we can look at those and sort of match those up with our list of targets and polygenic risk to try and identify potential repurposing opportunities. And I highlight one here for, I won't try and pronounce the drug, maybe I will, pegvisomant, and acromyalgi. And this is a growth hormone receptor antagonist, which is correlated with polygenic risk of type 2 diabetes. And that's interesting because there are separate studies looking at humans where there's a GHR deficiency, and they basically don't get type 2 diabetes. So potentially there's a link there. Potentially that drug may have utility in reducing risk of type 2 diabetes. These are things that are all in silico and need to be followed up on, but are very interesting findings that we can glean from polygenic risk, from using polygenic risk and combined with multi-omic data. Okay, so I'm running out of time here. So just to wrap up, really, ultimately, we're hoping that genomics is going to be rapidly feeding into healthcare, and that really needs to be its focus. It needs to be on trying to improve healthcare. We're trying to use we're trying to use the currently hidden information in our DNA to guide prevention and treatment of disease, and really, with the efforts of many interdisciplinary scientists and clinicians, as well as the efforts to educate the public. So me and I think many people who are working in the human genomics field really believe that it will be in the near future that this will be polygenic risk scores are going to be an important part of the healthcare that we all receive. So a few people to thank. I'd like to thank Sam Lambert and Gad Abraham, who've been working a lot on the polygenic score and clinical utility side. As well as Scott Ritchie, who's been doing a lot of the integrative molecular analyses, as well as many collaborators right around the world. And of course, can never forget the funders. So thanks for the opportunity to speak to you today.
Okay, well, thank you very much, Mike. So do we have any questions? We have a few minutes. So I'll start with one. I'm curious about the PRSRS, your standards initiative. So is that working well? So I guess the question is, you said that it was the organization of the data were poor, the things that you were able to get from different databases. Is it because there aren't standards, or there haven't yet, or up until recently, there haven't been clearly defined standards of what to include with publications? And that people, once the standards are clear, people will start following them? Or is there a real general reluctance to do that? Or is it, I suppose, also the possibility of a combination of the two, not having clear standards yet, and then also people reluctant to do it?
Yeah, so I think you're right. So I think it is a little bit of a combination of the two. As you know, things like sharing data, so data sharing polygenic scores. A lot of people come from different backgrounds. There are different sort of practices in different communities. Some people don't like to share, basically. So that being a key element of reproducible research and reporting standards, that is something that that really I think that the community still needs to get up to standard with, and that's why we've constructed the polygenic score catalog, or polygenic score catalog. There are other elements of reporting standards, like reporting about the ancestry of the participants, about the details of the statistical method, which are still kind of new, and I think we just need to raise awareness of that for the community. And I think this is one of the things that the polygenic risk score reporting standard has tried to lay out and really clarify for the community. I'll note here that it is sort of following on from a previous set of reporting standards, which I think were published in about 2011, so well before a lot of the kind of current era of polygenic risk scores. And that's the GRIP statement. So I think so to address the first part of your question, which was about were there other standards? So that is another standard which existed, but which was developed really with a lot of epidemiology in mind. So it's a really kind of generic reporting standard, which I think the community didn't really attach onto and didn't really appreciate, because it wasn't it just wasn't sort of up to date with what was going on. And so there was a real need for the community, which I think was recognized by not just the kind of European, the PGS catalog side, but also by the NIH clinical genome side, to update that and really get it back on the front page and get the community organized. Because now, as opposed to say in 2011, there really is an opportunity to translate into healthcare the polygenic scores that are being published, and to elevate kind of the level of research. So hopefully the PRSRS is going to do that.
Another question: how are polygenic scores inherited? If I have a high risk, what is the risk of my relatives?
Yeah, so I mean that's a great question. So polygenic score inheritance, off the top of my head, I probably can't give you. It's inherited. So it's a collection of variants, right, that are sprinkled around the genome. So adjusting for recombination and all that sort of thing, you inherit your polygenic risk from your mother and your father, right? And it's not perfectly like the average of the polygenic risk, and that can vary. And also there's various caveats to that as well, especially when you get admixed individuals such as myself, where you've just got different different I don't want to say genomic backgrounds, because that term's not very specific, but it can get quite complicated when you have multi-ancestry. So the point is that yes, it's inherited, but I think really we haven't we don't necessarily have like, I think the tools at the moment to take advantage of those inheritance patterns in the clinic. Really, we start from every individual as an individual, and we use population-based random samples of the population, their genetics, and hopefully linked health data to place that index individual in a distribution from a population, right? So you're in like the nth percentile, right? The 50th percentile, the 95th percentile, the 99th percentile of, individuals from Northern Europe or individuals of, Eastern European ancestry, et cetera. So that tends to be how we model that.
Next question. In the case of the PRS protein study, the Ritchie et al. Study, have you looked at the reverse to implicate proteins if their pQTLs are being disease associated? And if so, what's the overlap of the PRS correlated proteins?
So in terms of pQTLs in association with disease in the interval study, there is no power to detect those associations in the samples, which is roughly 3,500 or so individuals. And we're talking about a few dozen incident events. So there's really no power to look at that. When you look at whether they're associated with whether pQTLs are associated with disease, that is the case, and there's a separate study that's looked at that, Benson et al. In Nature 2018, which you can have a look at. When it comes to the PRS, so sorry, Keith, what was the last part of that question?
Oh, back to it. What's the overlap of the PRS correlated proteins?
Right. Okay. So I think that's implying. So that's then linking not just the pQTLs but the protein levels potentially, right, with disease. I think that's implying like Mendelian randomization. So I'm gonna interpret it that way. What we see and what we think. So this is all kind of still developing at the moment. Is that the two approaches of using polygenic scores to kind of validate or identify targets, and the use of Mendelian randomization are complementary. So there are some proteins. So I think roughly 2 of the polygenic score associated proteins that were associated with, I think, type 2 diabetes were also, when you use the pQTLs for those proteins and do a Mendelian randomization, they were also had a significant causal effect on type 2 diabetes. But when you look at actually the proteins that the polygenic score is saying are associated with or are mediators on disease, they actually, by and large, do not have the genetic instruments in order to perform Mendelian randomization. So, I think there's this interesting, not a dichotomy, but there's certainly a complementarity there of not every protein has genetic instruments or sufficient genetic instruments at least to do a Mendelian randomization, but they do seem to be those proteins that do seem to have polygenic score associations, and themselves, incident, are the protein levels themselves are associated with the incident disease. So what that means, we're still trying to tease apart, but the two approaches seem to be complementary, is the best I can say at this time.
Another question. You were talking about the need to standardize the reporting of PRS. However, these days, many of those scores are computed by some machine learning black box method or Bayesian method that does not necessarily release p-values. Do you have suggestions on how to standardize frequentist or non-frequentist PRS reporting?
Oh yeah, so it's a good question. I think what's important here though is to separate out the modeling itself from the score. So the score, that is the SNPs, the effect alleles, the weights, all of that comes out the other end, right, of the modeling. That's the thing, right, which you use in downstream studies, downstream as the sort of second stage of statistical modeling, right, on an external validation cohort, and then you sort of try and quantify the analytic or clinical validity of that score in a separate data set. And it's the provision of that score which was not happening. The modeling that went into developing those variants and those weights, we've not really dug into. I suppose there's so many different ways, like you know, right, like the machine learning sort of black box. You've got all sorts of hyperparameter tuning for deep neural networks. You've got differences between frequentist p-values and selecting features based on that versus Bayes factors, or so there's all sorts of. So we have not moved into that space. It's very much that I think that's very much a different thing that needs to be looked at, just because the expertise that is necessary there, and I guess the question really of the analytic reproducibility of the score is largely separate from the reporting that needs to happen for the score itself to have potential clinical translation potential. So yeah, I guess I would separate those two things. The first part is certainly valuable. The more that you can sort of provide information you can provide on the method and how it's developed and what the potential modes of failure are for that polygenic score, the better. But really the downstream part of once you've got the SNPs and weights, those need to be provided, and all sorts of downstream modeling metrics and methods need to be reported on. That's really what the PRSRS is focused on.
Okay, well, I think that's all the time we have for questions. I think we need to move on to the next presentation. So thank you very much. If there are a couple questions on the question and answer function in the Zoom seminar, so if you could take a look at those, or if people have additional questions, they can ask them through that particular that particular Zoom function. So again, thanks very much for your presentation.
Thank you.
Our final presentation today is actually going to be given by two people coming from the Department of Health and Health Sciences and Technology in the Institute of Translational Medicine at the ETH Zürich. The first person to share the Zoom link is going to be Kelly Ormond, and the second person, once Kelly is finished, is Effy Vayena. So thank you very much for being available to present today. We look forward to your talk.
Great, thanks, Keith. As Keith said, Effy and I are going to share this talk, and I'm going to begin by talking through the story of how secondary findings, reporting, and the lists really evolved over the past decade. And then she's going to spend a lot of time focusing on the underlying ethics and policy issues, and in particular, some of those that are relevant here in Switzerland. And I will disclose to those of you who don't know me that I'm very new to Switzerland, so I'm coming with the bias of having been in America throughout the development of all of this work. So if you think back a dozen years, 2010, 2011, 2012, at least in the United States, we were seeing the beginning of offering clinical exome sequencing, which really broadened our ability to make diagnoses. But in doing so, we moved beyond these top two categories here, where you were really only receiving results that were specifically related to your differential diagnosis and the clinical phenotype for which you were doing the testing. And we had the opportunity to receive lots and lots more information. And I think Michael really set the stage nicely by talking about how many clinical uncertainties we have just within polygenic risk scores. You can imagine if you then generalize that to other disease genes that are unrelated to the phenotype, carrier status, pharmacogenomics. We had a lot of situations where we were worried that we were going to receive information through the course of exome testing that might be unexpected to patients. And of course, for anyone who works in genetic medicine, you know that incidental findings are not new. We have had them for the eternity of our ability to receive genetic testing results. And some of these are about family relationships, and some of these are about conditions or prognoses that are somewhat unexpected. And this happens really across all types of testing. But nonetheless, this made everybody very nervous about how we were going to handle it. And the other challenge that was happening, at least in the United States at the time, where our clinical testing was driven by companies and academic labs, was that nobody had the same approach. So depending on what laboratory your clinical service chose to offer that test from, you might end up with a completely different list of things being reported. So this slide was one that I made probably almost 10 years ago now. People had differences in how they were asking for informed consent, differences in terms of what results they were returning, and differences even within some of these categories in what conditions were included. So you might send your sample to Baylor and get back, let's say, 50 different conditions that were considered to be medically actionable. And if you send it to a different laboratory, maybe you'd get 20, maybe you'd get 70, and they wouldn't always be the same. So from the clinical perspective, the real challenge was, how would a patient or their provider ever know that something had been covered? And so in it was probably 2011, Robert Green and Les Biesecker put together an ACMG work group, which I was a part of, that came up with this first recommendation, which was published in 2013. And really, when we think about what was going on there, our first question was, is there any list of conditions that we feel should definitely be reported when an exome or a genome sequence is done? And if so, how do we define what that list is? And the notion here was, we wanted it to be a minimum bar so that no matter what laboratory you got your sequencing done at, you would know that at least this list was included there. And labs had the opportunity to go above and beyond depending on their feelings about what should be included beyond that list, but it really was meant to be kind of the bottom of the list. And the criteria, we've tried very hard to think about things that had a clear phenotype, that the pathogenic variants were going to be detectable. So for example, triplet repeat diseases were not included off the bat and still aren't, because those weren't things that were going to be easy to detect through this type of sequencing. We really did want to pick conditions where there was a serious medical phenotype and that there was something you could do about that. So this notion of medical actionability. So when you put it on a slide like this, it seems like, oh, sure, that should be an easy process. We should be able to come up with that list easily and all agree on it. And I will tell you, and you've all probably lived through this, it's not easy to define any of these things. And we've come a long way in terms of trying to figure out what we do when we're thinking of defining these, but there's still a lot of areas of disagreement. So from the ethics standpoint, and again, this is just to get us started. I know that Effy's going to spend most of her 15 minutes talking about this. The notion that underlies the recommendations just in general is that there would be some benefit to individuals in receiving this information. It was really an opportunistic sort of screening in the sense that if someone was having an exome for a medical reason, we could easily obtain this information and it wouldn't necessarily change the cost or the testing samples or things like that. So it really could potentially benefit these individuals and their families. We were trying to minimize the risk of harm by focusing on things that we had clear analytic validity, clear clinical validity, clinical utility, and some management guidance. And the idea was that most people would not have the opportunity to receive this information otherwise. So it was recommended. One thing that was quite specific to the way that the US insurance system works was that it was originally recommended to be obligatory because otherwise there was a lot of concern that the insurance companies wouldn't pay for this part of the testing. Okay, and then the autonomy, I'm going to leave that out there because I know that's something Effy's going to talk a lot about. So you probably can't read this even on your screens, but the original ACMG list came up with 56 genes that covered 24 conditions, and the vast majority of them were cancer conditions or cardiovascular conditions that had some sort of risk for sudden death. So that list sort of stood in 2013, and not surprisingly, there was a huge controversial outpouring of concern in large part around the way that the autonomy issues and the decision-making was set up. But there were a number of papers. Again, I know that Effy's going to talk about many of these issues as she goes through her information. But the one that I do want to talk about here was the terminology of incidental findings versus secondary findings. And this was something that the original committee did debate. We started off with incidental because we really felt like the terminology of secondary connoted that they were somehow less important to the medical information for that patient. But I think many of those editorials and commentary papers talked about the notion that if you are looking for them on purpose, they are not really incidental. It's not like you just tripped over it while you were doing some filtering for the particular primary indication that you were looking for. So in 2013, the President's Commission came out with some clear definitions, and these are the ones that you'll find used in all of the papers that come out of the US. There's still quite a bit of difference in language across countries these days, but what we landed on was that the secondary finding terminology would be used within the US to look at things that are actively sought if they're not the primary target. These are also called opportunistic screenings, and I think that's the language that the European Society uses. And then incidental findings can be both anticipatable and unanticipatable, but those are things that you really wouldn't be looking for or searching for purposefully. And so we ended up in this place where there was quite a debate going on between people who are really seen as genetic libertarians. So these are individuals who really feel like this is the patient's data. We should give them everything that we have. We shouldn't worry about it. Versus the genetic empiricists who were more conservative and really felt like we should only return data that is going to have clinical significance and that we know enough about so that we're not burdening people with data that doesn't mean much. The recommendations kind of fell in the middle at this point, but I think this kind of characterizes the ongoing debate that many people have at this point. So since then, what's happened? Well, one of the first things that happened is that we've gotten better at having concrete ways to define clinical or medical actionability. And this is a paper that came out of some of the ClinGen work in the actionability work group that Katrina Goddard leads. And there's been a second version of this since, but this group really tried to define what do we mean when we say medically actionable, and how do we really think about that, and within different risk categories. So you can see in this table that they came up with categories of the severity, the likelihood of disease, the effectiveness of specific interventions, and the nature of them. So how burdensome are they? And one of the really interesting things is that they then went through the ACMG secondary findings list at that time, and you can see some of these scores, but one thing I really want to point out here on the bottom, so this is Putz-Jaeger, and you can see that there's a huge variation in scores within some of these conditions. So for example, colon cancer surveillance might get an 11 with good scores and good data behind it. Breast cancer surveillance and chemoprevention might also be fairly well scored, but you get down into ovarian cancer, risk-reducing surgery and surveillance, and those numbers aren't that great. And so even within a single condition, there can be some parts of the prognosis where we have good medical actionability and others where it's really simply not so good. And so these were some of the lower scored conditions. And you can see those are hypertrophic cardiomyopathies, FAP, and Fabry disease. So 2016 rolls around and the committee, which I was still on at that point, releases a new version of this. They increase the number of genes to 59 by adding 4 genes, which you can see indicated in blue here, and removing 1 gene from that list. Still mostly cancer and cardiovascular genes. And since that time, there have been a lot of studies. I've chosen one recent study from the Netherlands that has looked at what % of the population has a variant in one of these genes. And you can see that most studies are showing between about 1% and 3% of the population. So it's not a huge number, but it's not a non-significant number when we're talking about the number of people who may have exome sequences. The other thing we've learned a lot more about is that other countries and organizations have begun to develop lists. And importantly, they are not the same as the ACMG lists. So every country has got its own process for how they're thinking about this. This is a paper that came out of France a couple of years ago and was focused only on reporting cancer genes when sequencing is done. And the thing that I want to point out here is if you look at this Venn diagram, you can see the overlap. We have the ACMG first version of the secondary findings list, and then the second version, and then this was the list from France. And so there's a tremendous amount of difference between these two lists, even from well-represented professional groups that have been quite thoughtful about this. So we're continuing to see that people can't quite agree on what genes should be included and why. The other thing that we've had a lot of literature on, and again, I know Effy will speak to this a little bit, is how do patients respond to receiving this information? So this was a paper that was published in the European Journal of Medical Genetics about a year and a half ago from a group in France that did a systematic literature review. They found 39 studies that covered both participants and healthcare provider views. So it's a really nice summary of how people feel in terms of do they desire these findings and how do they respond afterwards. This is an example of one of those papers also published in the European Journal of Medical Genetics. And what I want to draw your attention to is the reactions here. This was a small study. 10 people out of about 2,500 tests had secondary findings. You can see most people did not have significant anxiety. A couple felt surprised or overwhelmed. One said they would have preferred not to know this. Most people said they felt prepared, although I always think it's interesting, despite the consent processes, we have many people say, I didn't remember I was going to get this information. I didn't realize it. And one person reports feeling pressured to receive it, which I think is why we worry so much about the consent process here. And then in terms of follow-up with these recommendations, you can see most people did intend to follow up in terms of the screening that was recommended to them, and most people felt satisfied. And this table really goes through for each patient what sort of finding they had. You can see, do you think you were sufficiently prepared? Did you have the necessary information to understand it? Yes, 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 yes. Everyone did. Do you remember what you learned about this incidental finding? Have you started the follow-up? So there's a lot of good data that people really are responding positively to this information. Despite this, the European Society of Human Genetics remains quite cautious and recommends that we continue to be as targeted as possible in the genetic testing that we order. They feel that opportunistic genetic screening really requires a strong burden of benefit. So just like the public health models that we're talking about, like Wilson and Younger from the 1960s, really needing to show that we're benefiting people if we're going to go out of our way to give this information, and that there's still a lot of uncertainty. And again, Michael's talk, I think, really illustrated that with polygenic risk scores. The same is true when we're talking about these dominant Mendelian genes. So I'm going to wrap up by talking about this paper that came out just a few days ago. The ACMG did release their 3.0 version of this list. And it's a nicely put together document that lists all of the genes that they have. They have changed a little bit of language now to use pathogenic and likely pathogenic, finally. And they have a nice table in the supplementary information. So any of you who are working in a laboratory, you can actually sort it really nicely. The things that are new on this list, they've added 3 cancer genes. 2 were related to pheochromocytoma and paraganglioma. One was POLB2, which is a hereditary breast cancer gene. All 3 of these are also listed on that French paper that I showed you a little while ago. They've added 4 more genes related to cardiovascular phenotypes, 2 metabolic conditions, biotinidase disease and Pompe disease, and then a handful of other conditions that have been included by many clinical laboratories in the US. So 2 genes for HHT, hereditary hemorrhagic telangiectasia, 1 for hereditary hemochromatosis. This is just the C282Y homozygotes, 1 gene for MODY3, which is a diabetes gene, and then RPE65, which is a retinopathy for which gene therapy treatments are available. They also had a really interesting table in this paper where they talked about genes that had been nominated by people for inclusion and why they chose not to include them. Some of those are technical and laboratory issues, so it might be hard to detect particular pathogenic variants in these genes, either because of pseudogenes or the type of mutation that's happening. Penetrance issues and management concerns were the main reasons. So these these may end up ultimately down the line being added to the list. That's something that now at this point, they're saying they're going to monitor this on a yearly basis. So I'm going to pass it over to Effy now, who's going to elaborate on the ethical and policy issues, and in particular, some of the work that the SPHN is doing. Right.
Thank you, Kelly, very much. All right, so I'm just going to, and thank you for the invitation to be here and the opportunity to share the talk with Kelly as well. I'm going to share my screen and move over to this. Oops, that's further down. Sorry about that. Right here. All right, so moving over from the context that Kelly presented, that was primarily in the area and the sort of space of clinical genetics, I will report to you some work that we did here in Switzerland in looking at what and how and why should we be returning to participants. Not to patients, but to research participants. Just by way of introduction, this is work that was conducted in the context of the Swiss Personalized Health Network. This is a network that's been in operation here in Switzerland for the last few years with the aim to facilitate data sharing and access, including also genomic data. This is, although distinct, not extremely different, at least in the vision or in the hope of other programs that have been developed over the years around the world, and they were mentioned already today.
Effy, sorry, we cannot see your screen or your slides.
Oh, I'm sorry. Okay, hold on a second. I'm trying this again. All right, so how about now?
Perfect, thank you.
Oh, I'm sorry about that. Okay. So I was right here as referring to the Swiss Personalized Health Network and sort of in parallel development to other international developments in this area. So this program was in 2017, early on in its inception, was based on an ethical framework which our group, I chair the, as a disclaimer as well, the ethical, legal and societal implications group of this network, a framework for responsible data processing that included various types of data, including genomic data. And the foundations of this framework were these 4 principles that are familiar to all of those in working in the area, either a clinical area or research: respect for persons, privacy, data fairness, and accountability. And on the basis of this very fundamental piece of work, later on the LC advisory group was asked to develop guidance about how to responsibly handle the return of results to individual participants that took part in studies that have been funded by the Swiss Personalized Health Network. And the report, the detailed information of how we got to work on this project and what we did and how we concluded is in this paper that was published recently. So let me go to some of the specifics of that work. One of the things that we also had to deal with was this concept of what is medically relevant research findings, the medical procedures that that they look for primary results, the secondary results, discoveries that are beyond the primary endpoint. And Kelly already mentioned that if you see on this table to the right, this is the definitions we used. And we moved quickly to the concept of secondary findings for reasons that also Kelly mentioned earlier on. Also, though, because the nomenclature here in Europe favored secondary to incidental very early on. So the question questions we are addressing is whether in the research context we should be returning primary findings, being those that the endpoints of the research product, or secondary findings, neither or both. And for that, we needed to come up with some sort of concept of actionability, the kinds of primary or secondary findings that can be used to guide clinical and personal decision-making, and the medically relevant findings, a separate category or a similar category, that again can be used to guide medical and clinical decision-making and treatment. So we make that distinction. I'll come back to this a little later as well. So the differentiation that we used across in the context of our work, that's in the clinical, we have the clinical context where a patient comes in, and there we might have secondary findings. So the question there would be, should we do that, under what conditions, the stuff that Kelly talked about. But in the research context, participants they're not necessarily patients, although we will come to the fact that maybe they're increasingly patients as well. So in the research context, should these genetic research results, primary or secondary, be reported? And if they were to be reported, what are the conditions under which we're going to do that? The distinction is relevant because typically we treat research and clinical research, or biomedical research and medical care, differently. And we treated differently, not only from the point of view of ethics and bioethics, which is traditionally has done that, but also from the point of view of the law. The primary responsibility in the clinical context is to take care of the patient, to provide the best care, and the focus is the patient. In research, the focus is the production of generalizable knowledge without, of course, harming the participant and respecting their rights. But these two objectives differentiate a lot the obligations, the responsibilities, and of course, this has implications on what's happening with the return of those kinds of results. Now, we have seen over the years that this clear demarcation is changing somewhat. Not only we have improvement in terms of the empowerment of the patients and more proactive communications that kind of break some of the fixed boundaries that we have put in place. We do have also different public expectations about the kind of blurry boundaries that we see between research and clinical practice, especially in the context of genetics. And with the declining costs of genomic sequencing, we're seeing this blurring between clinic and research becoming increasingly the space where we're moving in. So when we have this blurred space and kind of overlapping space, which principles really and which, let's say, guidance should we apply? The one that we typically use in clinical practice or the one that we typically use in research? So in thinking about the blurred space and starting from the point that we might have some sort of obligation to return something to the research participants, here are some of the reasons we considered in terms of what actually influences the decision to report results. And logically, you can see that they sort of make sense: the severity of conditions, the inheritance patterns, the time frame or the onset of the disease, significance of those risks for relatives, age of the participant, but very importantly, and the one that in the literature seems to be getting the most attention, obviously, is the clinical utility, which goes back into what we can do with the findings in terms of providing medical care or prevention. So the three primary concerns is the causal correlation between the finding and onset of disease, clinical utility, do we have the means to treat the condition or do we have means to prevent the condition? And also an increasingly important consideration with personal utility. Does receiving these kinds of this type of relevant information will be useful for the person, irrespective of whether we have a treatment or prevention available? That goes into the space, of course, of people making life decisions around some of those of these types of information. Now, when we're looking at practically, what is the right thing to do? Again, we look at the responsibilities, as I said, from the demarcation earlier, the physician to the participant, or the physician to the patient, or the researcher to the participant. Physicians, they do have the duty to promote the well-being of their patients. They do have also their duty to rescue them. They have issues of confidentiality, and they do have to respect the patient's autonomy. We've been through those also improvements of understanding what it means to respect people's autonomy, patients' autonomy, and their choices they can make. And these kinds of duties and ethical the duties that derive from those principles, they've been captured in various documents, from the Hippocratic Oath to the World Medical Association's declarations, and a number of other national guidelines and recommendations. When we're looking at the researcher's responsibility to participants, initially, of course, as the researchers may come across the kinds of information that could help rescue somebody, we see that we can raise there a duty to rescue. If it is possible, then the question is, is there a duty generated? And when this is an easy rescue, the rule of easy rescue, it seems that most agree that we can speak of that duty of the researchers. So through that, we have an ancillary care duty to research participants, something that's clearly up here in the physician responsibility. And up until recently, it wasn't really obvious that it's coming down to this. It spills over, let's say, to this context. We also have some other concerns, like that of reciprocity, as participants take always some sort of risk in participating in research, or trust the research enterprise, whether we owe them something and in return. And if what we owe them back is to inform them about things that perhaps have some meaning for them. And that links to this issue of autonomy and the principle of autonomy, particularly in the way that autonomy is understood as your right to be the author of your life, but on the condition that you have adequate information so you can make decisions for your life as a patient or as a research participant, or whoever that is. So this concept of autonomy and the requirement for others to respect autonomy of others creates also here some sort of responsibility to provide people with information that would allow them to make to be fully autonomous because of the weight this kind of information may have for their lives. So reciprocity and respect are the two, let's say, driving forces here that make bring kind of more weight into returning results, irrespective of whether they're primary or secondary to research participants under certain conditions. One thing in the ethical considerations that's been highly discussed, and it's in everybody's mind, is that, of course, people also have a right not to know, and that is inscribed in all sorts of guidance and laws in many countries. And so participants as autonomous, have the autonomous, can make the autonomous choice not to be informed about their genetic information. Now, questions that have been, we've dealt with in the past and we're still dealing with is, can this right be overridden by a professional's duty to act? So there is a professional duty to act in the best medical interest of participants, but how powerful is this right in overriding one's right not to know? Particularly, all of that comes up only in the case of a serious but preventable medical condition. And it looks like the jury is still out as to what we're going to do about how we're going to resolve that, because, as you can imagine, it's quite a challenging issue. And here we do have some considerations going in sort of redefining what autonomy is. And again, autonomy based on the fact that not in general, I don't want to know anything about this, but maybe autonomy is the way in which we make decisions when valuable information is available, and then we choose through a range of valuable options. So participant autonomy and what it really means actually was seen in a new light through these debates about what do we owe to participants? Can we override certain of their rights, and how we're going to do that? If another consideration that has seriously come up in these discussions and the development of recommendations is what is the role of and the health of third parties. For example, if information through a person could lead to different choices or prevention or screening or even specific reproductive choices in this person's relatives. And we do have cases, including actually legal cases, that touch upon the difficulty of these kinds of questions in relation to relatives. When we developed the recommendations, we always, of course, had to look into the Swiss legal context and what is possible to do here. Here in Switzerland, we have at least two acts that are very strictly very related to these questions. One is the Human Research Act, and the other is the Human Genetic Testing Act. So according to the Human Research Act, the persons who take part in research or clinical trials here are entitled to be informed. And that kind of information should be given in an appropriate manner. But of course, at the same time, in the Human Genetic Testing Act, we have the point about the right not to know. So every person has also the right legally to refuse to receive information about their genetic status. And so the question, again, is there any way to reconcile those and respect both of those, which clearly derive from the ethical principles that I mentioned earlier. So in our recommendations, we kind of navigate the principles and the legal context. And in the general overview, what we ended up doing is that we emphasize that the reporting of any genetic findings of medical relevant participant is advisable. We present some options. This should be presented. The options should be presented in the during the informed consent process, because that can allow people to make certain choices ahead of time and not later on. Looking at the findings, again, emphasis is given to those for which there's preventive or therapeutic measures, and the genetic variants linked to conditions that are not preventable or curable. Of course, they are treated differently. So the decisions about genetic findings that we suggest here is severity of the detected condition, the existence of clinical treatment of prevention, obviously, and the relevance of the variant for descendants of the participant, and related to that, personal utility. In our recommendations, we did not come up with a list of genes as others have done, and the National Society here in Switzerland has not produced such a list. In fact, there's quite a lot of resistance in coming up with a certain list. Kelly showed us how we're still struggling with agreeing in what should be reported and what not. And so, at least in our recommendations, we suggested that we do not have a fixed list, but this is a case by case sort of decision on the basis of the projects. Another thing that was important, at least in our set of recommendations, was the suggestion to have multidisciplinary expert panels to support the decision making for when a project in the conceptions of the project in terms of what should be returned. Instead of the research team alone deciding what should be returned to participants, have a broader kind of consultation to make that determination. And we've seen also that this seems to be the case, the approach that other countries are taking. As I mentioned, the case-by-case decisions than predetermined list of genes, that was a decision that was made on the kind of pragmatic decision on the national take from a medical geneticist here. It was a bit of a contentious issue, but one that we took the pragmatic approach of the country. Now, another thing I want to raise quickly is the implications for informed consent and privacy, in particularly for insurance. In Switzerland, again, if people are aware of genetic mutations for certain forms and potential conditions, they need to report those for when they claim they want to obtain life insurance, and any implication on insurance from results that will be given back to people, so people will be aware of certain things and they may have to disclose, can have consequences for them in the future. And that's a really important point that has to be addressed at the time of consent, and could also serve as a deterrent for people's participation. That's not just a unique situation in Switzerland, but that's something that I think is worthy of attention. Hold on a second. There's one more comment, that one more issue that we addressed here in the context of consent and form of reconciliation of the different approaches that and different arguments. There are recommendations also concluded that if people were given if a project, in a particular project, the decision was to return certain results and participants did not want to receive results, the participants should be excluded from the research project. That's one that we can talk about if you'd like in the Q&A. It might be a little different from other places, but there is the justification for that is to reconcile the right not to know and the duties and obligations of research participants on the other side. So in conclusion, I think what we're seeing is, and in both, not only in the research context, but the clinical context, what I call learning to live with genetic information. Clearly, we have more of a tendency to return results, whether these are primary in research and secondary, or secondary in the clinical context. We also see the necessity to understand the attitudes and needs of all stakeholders, and that's something we're seeing in many countries, and clearly to deliberate with the stakeholders in order to reach some sort of conclusion and agreement. We're seeing these models, that's what we practice here in Switzerland, but we're seeing also these models in other places and with other guidance that is emerging. Thank you, and I will stop sharing here.
Okay, so we have time for a few questions. Okay, so I'll jump in right away. So the discussion, or the dynamic of the discussion between, I think, Kelly, you called them the libertarians and the empiricists, giving people everything, giving people just a very directed set or view of the results. So it may be too early to know, but I guess maybe what I'd like to know what your guess is. So after living through a health emergency for 18 months, although it's an infectious disease health, it's not necessarily a genetic, the genetics aren't necessarily clear. So how do you think this is going to impact that discussion? Is it going to move people to the discussion in one way or another?
That's a great question. I think people have learned a lot more personally about how they deal with risk and uncertainty, for better or worse. And so that may mean that people are more comfortable with getting information that's uncertain. And it may also mean that providers are more comfortable explaining some of the uncertainties that we have. I think often as clinicians, we have a tendency to talk about medical findings in a sense that makes patients think it's very clear-cut and all sorted. And we know it's not. So it may shift things a little bit, but it may also be that people feel like, oh, you know, that's too much. I realized that I didn't do well with it during all the COVID time, and I just want you to shut it down. My own views, I think, are a little biased because I come from the Silicon Valley, where everyone thinks that everyone should have all their data and be able to play with it. And I don't personally think that, but that's the environment that I've just come from.
Great. Okay, another question. Effy, you mentioned third-party interests, for example, relatives of focal patients, and that this is a difficult question to solve. Do you know of examples of consent forms that take this point into account? And what are some of the specific complexities arising in the context of balancing goods and interests here?
Thank you. Also, yeah, that's a great question too. And I think this could be also slightly different from in the clinical and research context. Kelly can speak about the clinical, but for the research context, I think what we're seeing is encouragement of encouraging the participants to talk to their relatives about what's going to happen and decisions they've been made. Typically, the consent form is about the the participant, the individual. We've seen some suggestions for family or relative type of approach in the form of an agreement, but this is more, I'm not so sure how this is practiced. I've seen that in the literature as a suggestion with good reasoning why we should do it, but I think the practice is still different, and it is that you try to explain to people that they ideally talk with their relatives, because the implications can be severe, or they can cause them quite a lot of trouble. So it's a very delicate issue. Legally, I think we haven't resolved it. We still, in the clinical context, we have some cases like the one we had in Scotland, but in the practice, we're still practicing individual consent and just try to talk to their people, so to speak. Kelly, I don't know if you've seen something in the clinical context that's different from that.
I agree with everything you said, and I don't think it's showing up actually on the consent forms. It is more a discussion point. But the other place I think it comes up is when you're doing genetic testing on a child and you discover a secondary finding in a gene that has mostly adult onset, and then really it's most impactful for the parents, and you have to sort of navigate how to disclose that, and it gets complicated clinically. Yeah.
May I ask a question? If I
Please
can.
do.
Instead of writing it, I can just say, since I'm live. So I'm just going to be a little bit provocative here, but not surprising, given me. There's a discussion that is actually being made in more, I could say, sometimes informal settings, where several of us, including myself, believe that much of the discussion around what should and should not be communicated remains between experts in, let's say, humanities, let's say professors, academics, and the scientists that understand this information. And the public actually hasn't understood enough of what is at stake to ask the right questions and pose the right concerns. So that's my impression too. I believe that to some degree. So the question is, have we engaged the public properly so they fully understand what's at stake here? What are the problems and the risks and the issues, so that we fully understand the level of the concern? And just to add one thing, I actually believe that we're more concerned than the public is. And this is the thing that concerns me. Are we engineering concerns that actually the public doesn't have, at least not as strongly, leading to over legislation, over regulation that actually nobody at the end wants, and especially the stakeholders?
What a terrific question. I think that the point that you're raising and what I'm hearing here is, are we being too conservative? Are we creating these issues? And as genetic counselors, we've done this forever, right? We've been so conservative in worrying about the psychosocial outcomes. Maybe that people just aren't going to be that upset by this, and they're going to be grateful for this information, and we just need to educate them and roll with it. You know, I think the hard part is we don't know the answer to this yet, and we really do need to do some more studies and do a better job educating people, but also be open-minded, I think, because there can be people who really don't want to know this, and absolutely it can be a bad thing. So.
But also what we see from the studies, for example, in return of results, the majority of people want them. We see just a small percentage of people who don't want them. So it seems that a lot of the guidance is in order to take care of that smaller part that it's unlikely that they don't want it. So, but the majority, okay, I understand we cannot go with the voice of the majority, and we should be careful and, and respectful of everybody's individual wishes. But I agree with that. And I wanted to add one more point, and I don't think we're making incredible progress, Manolis, if that's in how do we deal with that, the issue you're just raising. I think we're also having another issue, which is very often when we introduce uncertainty to patients or research participants, they ask you back. There is a burden as well in asking them to make a decision with the limited information of context they may have. And so there, I think we got to be quite careful. On the one hand, empowering people, they have absolutely the right and they should have the information to make decisions, but there's a burden that comes with some of this as well. And there, I think we're not striking necessarily the right balance. I think sometimes they sort of case by case, talking to people or trying to educate, not to educate them, but to give them the right information of context is probably the way, but that becomes case by case. That is extremely time consuming and resource consuming. And I think it's not happening. So I'm kind of thinking some of this guidance takes care, it's like the Band-Aid for some of this bigger problem that we have trouble addressing. But I don't have a good solution, I admit.
I think the other challenge that comes up with what Effy talked about on the case-to-case basis is if you raise with the patient that we have possibly found something and we want to talk with you about whether or not you want to learn it, it is, I think, virtually impossible for that patient or research participant to then decline it, because they feel like if you found something and you're even bringing it up, it must be so important and you must understand it well enough that it would be meaningful to them. So I think that puts them in a bit of a dilemma as well.
Well, good. There's no questions in the Q&A. No one sending me a chat asking with a question. So thank you, Effy and Kelly, for a really great combined presentation. I'd also like to thank all the speakers this morning for their presentations. It was striking in the range of topics that were described and discussed. The one thing I think common to all of the presentations is the high quality of each and every one. So thanks very much to everybody for participating today.